So good afternoon, everybody. We are back here. Professor Geoff Goodhill is with us to respond or to answer the question you might have regarding his presentation this morning. So we have a few questions for the moment, but I hope people will continue asking while he's responding. So let's try to start inviting the first person in the list, Shabika Rastogi. So let me invite him on the screen. I don't know if he's not there or she is not there because it might take some time. I think it takes some time. She, okay, I think it takes some time. It's there. It takes a moment. Okay, she's there.
Yeah.
Thank you.
So hi.
Hi.
Thanks for the nice talk. So my question was how the axon, as you told in your presentation, that axon guidance is affected by the range, it's sensitive to the range of concentrations. It is there only for a narrow range of concentration. So my question is, how is it affected by the change in concentration? For example, if concentrations do change due to thermal fluctuations or due to stochastic noise in the brain. So how will it change the axon guidance? So will it affect it in any way, or is it robust?
Right, so those are two slightly different scales. Before I talked about the model which predicts the narrow range of sensitivity to gradients, we talked about modeling the thermal fluctuations and the receptor binding noise, and so that gives you uncertainty in your estimate of concentration of a few percent for typical parameters, in terms of the concentration range for effective guidance, that's about 2 orders of magnitude. So that's a very different sort of range. And I mean, intuitively, what's going on there is just that at very low concentrations, you don't have enough receptors bound to get a reliable signal. And at high concentrations, essentially all the receptors are bound and you can't get a reliable signal. And so you have about 100-fold to play with in there.
So it's robust to the range to the fluctuations in the concentrations, right?
Well, as I say, I presented those calculations, which tells you exactly how those fluctuations affect the sensitivity. So those calculations tell you the fractional uncertainty in your estimate of a concentration.
Okay.
And so if you compare that between the two sides of the growth cone, that gives you a sort of a sense of what the minimum gradient is to be robustly detected. But as I say, that's a slightly different issue from the complete range of concentration.
So does this range of concentration which you talked about for the reliable axon guidance, so does it change over the course of development, or does it remain the same?
Right, so this, I mean, I didn't really emphasize this in the talk, but there's nothing in that model which actually has anything to do with axon guidance. It's just a model of a small sensing device. And so actually that applies to any sensing device, be it a growth cone or anything that's using a spatial sensing mechanism. And so from that point of view, no, those constraints don't change over development. Now, what might change over development is the receptors that are expressed on the growth cone. So because that curve shifts with the dissociation constant. So it's roughly centered on the dissociation constant, the receptor ligand interaction. So if you express different receptors and respond to different ligands, then you'll have a different range of, or so if you have the same ligand, but you you have a receptor which has a very different dissociation constant, then you could use that as a way of extending the concentration range if you down-regulated one set of receptors and up-regulated another.
Okay. Okay. Yeah. Okay. Thank you.
Okay. Thank you very much,
Thank you.
Erika. Thank you.
Yeah.
Sorry. We have Tom Burns, which is around, so he has several questions. So I will invite him also to be on the screen. Yeah, it's not immediate. It takes some time. Now it's there. Hey.
Hi, Tom.
I might ask the one question.
The sound is not very good.
So I'll just ask my own questions if it's okay. Yeah. So two questions I had was, does zebrafish display social behaviors at different levels of complexity? And if so, do wild-type zebrafish, are they better at maybe reproducing the more complex behaviors than the knockout or the Fragile X fish?
Right, so we've mostly focused just on the very early stages of social behavior, and we were following some nice work by Elena Driosti, who has developed this U-maze assay, where fish sort of swim between two arms of a maze. On one side, they can see a fellow fish, and on the other side, they can't see a fish. And the data I showed in the talk was, when they're looking at another fish, how they're responding to those movements. But there's also part of that experiment is also how much time they spend on that side of the chamber compared to the other side of the chamber, where there isn't a fish. And in fact, the Fragile X fish, actually, sort of counterintuitively, they actually spend more time on the side where there's another fish, because you might think that, sort of intuitively, you might think they'd be less social, but in fact, they seem to be more interested in other fish. They just are worse at responding to them than wild-type fish. So, in terms of so we haven't looked at adult fish, and of course, adult fish do have shoaling behavior and mating behavior and so on, but we haven't looked at that, so I can't say whether there be differences in it.
Yeah. And actually, it's interesting what you just mentioned. I wonder if that would even be a compensatory kind of mechanism, like staying near the other fish because they're maybe not good at the social interactions, so they try to increase their chances or something of having a good
Could be, yeah.
And another question I had was actually a bit related to the question you just had before. So I think in the model you showed for the growth cone, it was a 1D model. Was that correct? Like the growth cone itself was 1D.
Right. So there are a few different models there. So the
Okay.
you mean the Bayesian model?
Yeah.
Yeah. So that was 1D. Yeah. So we also extended it to 2 dimensions.
Right. Okay. And 3 dimensions, is that? Did you extend that as well?
Or? No, just the
Okay.
3 dimensions. I mean, there was nothing particularly interesting that happened in 2 dimensions compared to 1 dimension. I mean, it certainly didn't help us understand the experimental data we had.
Yeah. It seemed like a good fit, so I wasn't sure that it would necessarily improve. Yeah, but just curious to know if there was any difference.
So we had a paper in neural computation where we looked at the 2D case.
Yeah, and actually, have you also looked at sort of interactions between different ligands and different concentration gradients competing?
Yeah, so that's a great question. So that's a question of great interest in the biology, because we know there's often gradients that collaborate. And so what we considered was the issue of, say, they were collaborating in an optimal way, right? So again, that's in that same paper where we looked at the 2D case, that same neural computation paper. We looked at what would be the Bayes optimal strategy to combine information from different different ligands, and of course that depends on their concentrations, right? Because for very high concentrations and very low concentrations, they're not giving very reliable signals. So I, can't remember off the top of my head.
But I guess you've got to have sort of a perfect storm of these concentration gradients to get like the perfect or fastest growth.
Yeah. So I mean, one conclusion from that work was that having multiple gradients doesn't solve this problem of there being quite a narrow range of concentration.
Okay. Just one final question then. What hypotheses do you intend to be testing in the whole brain zebrafish imaging during sleep or 24-hour recordings?
Right. So there are some very nice computational models of sleep, and but the biological substrates of those are less clear. So things like the two-process model, and so by looking at whole brain activity, the hope is that we'll be able to really test where in the brain are the substrates of some of these models.
And I guess because you've also got the model, the SIVA, is it called, or
Silver.
Silver. Yeah. So you can look at silver-evoked and spontaneous activity across the whole brain, and see those different systems. Okay, cool.
Sure.
Yeah. Thanks very much.
Thanks, Tom.
Hi, Jeffrey. Maurizio de Pidella from the Basque Center of Applied Mathematics in Spain.
Hi.
A couple of questions. So, might sound a little bit mind-provoking. So, is there any glia in the zebrafish brain? And if so, what do we know about its role in the axogenesis and neurogenesis of these functional circuits?
Right. So there are certainly glia there. And I can't tell you very much about their role in neural development. That's sort of not something we've looked at. But if you're interested in glia, I would definitely refer you to a recent paper from Misha Ahrens at Janelia Farm. So Ahrens. And they found that glia play a very important role in certain kinds of larval zebrafish behavior. So this is a story that was recently published in Cell. But with regard to their role in neural development, that's just not a thing we've looked at. So I'm afraid I don't have much to say about that.
So then switching to some other things. So like you showed this interesting video about this clustering of activity in the tectum, right? So is this clustering somehow occurring in spontaneous conditions, or is it modulated by the actual movement of the zebrafish in some way, some cognitive task or some kind? Do we know about this modulation or the way this modulation could occur?
Right, so the tectum is upstream of this. And so in our preparation, the fish is completely embedded in agarose. People like Herman Sombre in Paris have looked at tail-free preparation. And they had a nice paper in Neuron in 2015 where they looked at spontaneous assemblers in the tectum. So they were just looking at one age, but they had this tail-free preparation, and they found that it was often the case that activity in the tectum was correlated with tail movements. So the idea is spontaneous activity in the tectum then drives the tail movement. It doesn't happen all the time, but some of the time.
So what about your Zebrinator model? You show these very interesting simulations. I didn't quite got if the model is 2D or 3D, in which you study essentially the perturbation of turbulence in the environment of the zebrafish. It's a recent work that you're saying it's in progress right now.
Yes.
So could you say a little bit about this model, if it's like in actual 3D environment, or you're studying?
Yeah, absolutely. So it is in a 3D environment. So I'll tell you the complete workflow. So we have all these movies which of zebrafish moving around, and so those movies are in 2D, but obviously we know the 3D shape of the fish, and so we take we do image processing on the movies to extract the backbone of the fish, and then we have a 3D model of the fish, and we make the 3D model behave in the way that the actual fish is behaving, and then we calculate the forces involved in that using this computational fluid dynamics software. So it is a 3D model. Obviously, I was just showing a 2D sort of cut through that simulation, but we know what's going on in 3 dimensions there.
And out of these simulations, did you find out there is a potential feedback from the perturbation of the environment on the actual sensory system of the zebrafish, so that essentially the fish is moving in a sort of optimized fashion or according to what is his own effect on the local environment?
Right. So
it might be a little bit far-fetched, but just ask.
Yeah, I mean, clearly the fish is getting feedback from the fluid flow because it has a lateral line system which is measuring fluid flow. I guess we can't say anything from our simulations about to what extent it's it's directly sensing that and using that to control its motion. I mean, the questions we're more focused on is the efficiency of different bout types, because there's the order of a dozen different types of movement it uses at this stage, and so we're interested in how it selects those different kinds of movements and whether the efficiency of those movements is helping to determine which ones it selects at different ages, because the relative efficiencies of different types of movement change with age as the fish grows.
Right. And the last question in this regard, and my last question in general, is: do you actually observe what we call FAPs, so fixed action patterns, perhaps in the zebrafish? Like, there is an evolvement about these FAPs in the zebrafish across different stages of development, or not?
Sorry, could you just say that again?
I'm saying, do we observe any kind of FAPs, so fixed action patterns, at the level of neural correlates in the zebrafish? And if so, is there like a development? Hi, Martin.
Hello.
And is there like a development in of these FAPs as the fish grows?
So, I mean, the fish movement essentially consists of these discrete bouts. So each bout, lasts 100 or 200 milliseconds and consists of a series of movements of the tail. And then at this age, those are separated by periods when the fish is not moving. And as I said, there's about, people have classified maybe a dozen. I mean, it also depends how finely you slice them, but roughly speaking, people agree there's about a dozen different types of bouts. And so far, we haven't, this is a level of detail which we didn't get to in this current biology paper that's just about to come out. But so far, we haven't seen fundamental changes in those stereotype movements. So at the moment, at least over the period we're looking at, they're fixed. I mean, if you look, other people have looked over, zebrafish going all the way up to adults, and I mean, the movements become a lot smoother. I mean, adult zebrafish, they don't move in these discrete bouts. They swim continuously.
Well,
so
I mean,
there's a trend.
The question here is not really to see perhaps the change in the movement, rather to change in the frequency of the usage of these movements.
Yeah, so that's not something. I mean, we have the data. We're working on it. Yeah, so to look at whether there's changes in the frequency of bout selection, the selection of different bouts, whether that changes with development, that's something we're working on at the moment.
Thank you. Thank you very much.
Hi.
I can hear Geoff Goodhill, but I cannot hear the moderator of the session.
Yeah, it was my fault. Sorry, I had my micro off. Sorry, Martin. Yeah, you can start now. Sorry.
So hi, I'm Martin Zapotocki from Prague. I work actually on modeling of axon growth and axon mechanics. So I know some of your work that you were discussing in the first part of the talk. Really, very nice work also to read about. I was looking forward to meeting you in Melbourne, but in 2 years, right? In 2 years. Today you talked about your work showing the fundamental limitations on growth cone navigation arising from biochemical signaling. And I'd like to ask a question about another possible source of limitations, which is the mechanics of the axon. Even if a reliable signal is generated in the growth cone, then the cytoskeleton in growth cone has to rearrange. Traction force has to be generated, which depends on having good enough adhesion to the substrate or to the extracellular matrix. There may be mechanical tension in the axon shaft behind the growth cone, which would basically make the axon continue to go straight. Okay, so there are these mechanical factors, and I'm wondering if there are situations, if you are aware of some situations, either in vivo or in vitro, in culture, where the mechanics could be a limitation. For example, in a strong gradient, maybe there is some limitation on the possible radius of curvature, or reacting to a weak gradient, probably the cytoskeleton has enough time really to rearrange. But reacting to strong gradients, maybe, for example, if you're moving in an environment where you cannot get a really good grip and have difficulty generating traction force, one could imagine that there would be inefficient turning, even if there is a very, very strong signal.
Right, so that's a very interesting set of questions. I think in terms of constraints on its ability to make a sharp turn, so there are certainly cases in vivo where axons do make quite sharp turns, and they can slow down to do that. But in terms of the exerting traction forces, then I think that's a really interesting area. It's not one that we've worked on, but people like Christian Franze at Cambridge have been very interested in that, and you probably know that work. And there's some evidence there may be gradients of stiffness in the developing brain, and axons prefer to grow on stiffer substrates than less stiff substrates, as I recall. And I think that's a really underexplored area, and so I think that's all I can say is I think that's a very interesting set of questions, but not one that we really have much to say about.
But you would say it's an interesting thing to maybe work on.
Definitely, yeah.
And just curious, do you know of any situations? Have you seen any situations where, for example, the growth cone would be slipping and not doing a smooth motion, but basically
right? So there's a
behavior during turning.
So there is definitely some work. I'm not sure about slipping, but there's definitely some very interesting work on the interaction of the growth cone with substrates of different stiffnesses, and how that varies with stiffness. I can't think of it off the top of my head, but maybe if you send me an email, I'd be happy to get back to you with some papers about that.
Sure. Great. Thank you very much.
Okay. Thank you.
Thank you, Martin. Let me see if we have more questions. And not for the moment, but I have a few very naive questions I want to ask you also. I remember you showed us a video with the axon growing. I think it was only one realization it was repeated, so I guess it was the only realization. But I can imagine that if you have many realizations, you would be able, with some machine learning tool, for instance, to predict the direction of the growth of the axon. Would that be possible or not? Or is too stochastic, the process,
The process is pretty stochastic. another problem is, so one of the movies I showed was it just growing across the bottom of a dish with no particular guidance cues. And that, I think in the talk I said that dish was glass. I think it's actually a plastic dish. And there's, a certain amount of texture on the bottom of the dish, which is just from the manufacturing. And related to what we were just talking about, the growth cone is sensitive to variations in the substrate. And so I think, that would be so the substrate is perturbing the growth cone's movement. So from that point of view, I think it'd be very hard to predict exactly what it's going to do next, because there's environmental cues which are very hard to control, which are helping to determine, the direction it grows in at each moment.
Mm-hmm. Okay.
So
I know what you mean. Then when you show the zebrafish, you say that when during development they become more precise in hunting, or would that be related to the necessity of the fish for more food? Since they are developing, they probably need more food. Do they improve because of
yes,
the
I should. Yeah, sorry, I should have said that in the talk. But so up to 5 days post-fertilization, they're basically living off their yolk sac. But then that starts to get depleted, and they need to catch their own prey. And so if you don't, so they can survive till about 7 or 8 days if you don't feed them. But starting from 5, they're starting to catch their own prey.
Okay. Well, just my last question, because Thomas is also entering now. Then when you talk about the learning rule and these patterns of spontaneous emission, you show that with learning rule you have some patterns that start to appear from this quite stochastic environment. That reminds me the idea of the polychronization of Eugene Isikievich, where he also had some spontaneous activity and adding some learning rules, you start to see some pattern that remains there. It's a kind of learning plasticity rule. Would that be more or less similar to that?
Yeah, our work is certainly related to that, and I can't remember all the details, but I do know that we cite that work in our paper. So so it's certainly related. I mean, we were very focused on trying to quantitatively explain some of the properties of our experimental data from the zebrafish.
Okay, thank you, Thomas. Your turn.
Hi. Sorry for taking you in completely different directions once again, but I'm really interested in the spin microscope technique to have a whole brain recording. I mean, that sounds absolutely fascinating. And I had 2 questions about that. So one of them is, what are the limitations on the kind of dyes you can use? I mean, could you do this with voltage sensitive dyes? But then also, what limits actually the update rate, which seemed for the whole brain, it's good, but 1 second or 1 hertz isn't.
Yeah. Okay. So there's a bunch of trade-offs here. So the basic technology is that you use a 1 photon or 2 photon laser. You turn it into a sheet and you send it through the brain perpendicular to the scope. Now you can do that at a frequency of the order of 100 hertz, right? But the thing is, for one plane, but you also want to capture the whole brain. And so this sort of 1 volume per second time scale comes from the fact that you're scanning the sheet through maybe the order of 50 planes. So you're only spending like 20 milliseconds on each plane.
Does that have to do with how long you need to excite, or does it have to do with that you can't move the mirrors any faster?
Well, so there's a combination of factors. You mean what's limiting it to 100 hertz? I mean, it certainly involves both of the things you mentioned. I mean, obviously, the less time you spend on each plane, the dimmer the signal is going to be. And also, you do get to the level of having some, there's different kinds of technologies you can use. I mean, what seems to be the way to do it at the moment, the way we use is use an electronically tunable lens, because you need to be moving the focus so that it's matching where the sheet is. And the early systems, some early systems had did that mechanically, but the objective of the microscope is pretty heavy, so you get all this bouncing. But now people tend to use this electrically tunable lens, so there isn't a moving part up there. With regard to the sensors, then we're using GCamp 6S, which is nuclear targeted and produces these very nicely well-defined neurons. A disadvantage of that is it's got a very long decay time of the order of several seconds. And so even if you could image a lot faster, it wouldn't necessarily help you very much with a sensor with those kind of connections.
No.
Now, so voltage sensitive dyes are certainly a very exciting area. There's some really fundamental problems one has to overcome with those. So one of which is that they're obviously present in the membrane, right? Because that's where the voltage is changing, and so that's a very small volume. So we're measuring calcium in the nucleus, but if you're just looking at the volume of the membrane, you don't have many fluorophore molecules there.
Also, it gets very
It's very dim.
Right.
Then also they seem to bleach at the moment. They seem to bleach very quickly.
Oh yeah.
And of course, if you want to actually use them, assuming they're sensitive to millisecond changes, then you have to be imaging at a sort of millisecond timescale in order to see that. So you've really got this, all these things just working all in the wrong direction, right? You need to be, you've got very few fluorophores, and you need to be capturing an image in the order of a millisecond. So, I think, eventually the technology will get there, but I think, and people are starting to do it, but it's certainly not a routine tool that many labs are using at the moment.
And so the frequency would go up if you just limit yourself to presumably less sheets, or
yes. So it's all a trade-off, right? You can get lots of neurons at slow temporal speed, or a few neurons much more rapidly.
Right. I get you. Yeah, very exciting. I like this stuff. It makes for very, very good movies as well, obviously.
Yeah.
Thank you very much.
Thank you. I can't hear you. I think you're still muted.
Yeah, I muted the micro just in case there are some guys. Now you hear me. Well, you responded all the questions. I think you were quite clear. Now, if you want, we can wait probably a few minutes. Just if someone else want to ask another question, we have still some time. But if not, we can end the session whenever you like.
I'm happy to wait another couple of minutes just to see if anybody else shows up.
Can you comment in the meantime? Can you comment in this where you found this these clusters of activity in the? Can you comment on that, because it looked like you have some cluster? How was more or less the synchronization between them? Were they synchronized in time, or what kind of dynamic?
Right. So that's a great question, and so one of the problems with that movie is that it's speeded up 15 times, and so there's lots of things that you see there which are not really there, because everything's being compressed in time. And so we actually, for typical Tectum, we might find, say, 5 or 6 assemblies, right? And they're spatially localized. And so we were very interested if there was any sort of temporal sequence. So on average, you might get an assembly event, every of the order of 1 minute. But that's already a sign that maybe you wouldn't expect to see much dependency between different assemblies. Obviously they're occurring sort of randomly, so sometimes they occur closer together than that. But we did do an analysis to check whether there's any systematic ordering in which the assemblies occur, and alas, the answer was no. I was really hoping there'd be something more interesting, but alas, they really seem to be occurring randomly.
Okay, good. Thank you for the response. Well, I think we don't have more questions, so let me thank you again, Geoff. It was great to have you on board, and I hope to see you soon. Thank you very much for the very interesting and nice talk.
Thanks again for inviting me.
Okay, bye-bye.
Bye.
All right. Hello, everybody. and we are back for the members meeting. And I'm not going to take any time, and our president Volker will start us off.
Hi everyone. So welcome to this year's OCNS members meeting. So you're obviously very welcome if you're a member, but you don't have to leave if you're not a member. This meeting is open for everyone. So it's if you are a member, you have the right to suggest additional agenda items. So if you want to bring anything up which is not on the agenda, please do so using Ask a Question, the button that you can see there on Crowdcast. So what we're going to do is I'll briefly report on our activities, and I'll give a quick report on the OCNS membership. That's then followed by a report from our treasurer, Leonid Rubczynski. Then Thomas Nowotny, as program committee chair, will give us his report, followed by Thomas Knösche, who will give a presentation about CNS 2021 next year. And at the end of the meeting, we present this year's mascot. That's the result of the mascot competition. And if there are any matters arising, so if you've suggested any other agenda items, we will talk about those then also. So as you know, the meeting was originally planned to take place in Australia, and then we had the lockdown situation, and we had to put in place with very short notice in online meetings. And we managed to do that with the help of an excellent web organizing committee. So the first thing I want to do really is to extend my thanks to the web organizing committee that volunteered. So we managed to recruit them from our membership, and they did an amazing job, I think. So I'd like to thank Shailesh Apukutan, Tom Byrne, Sharon Dora, Wei Chin, Soledad Gonzalo-Cogno, Rebecca Evans, Elena Stellaris, Katarina Zulsdorf, Raghavan Yanaki, and Anand Patak, who helped organizing social media, the web, answered your questions, and without them, this meeting would have really been impossible. And then I would also like to thank the OCNS directors who were involved in organizing this meeting. In particular, Anko Sinha, who did an amazing job, as always, as webmaster. So he's responsible for a lot of work behind the organization of CNS 2020 online. And I'd also like to thank Thomas Nowotny, who was the program chair. And I think the program committee managed to put on an amazing program this year as well. So I'm really impressed with the quality of the talks that I've seen. So just to briefly talk about some of our activities, the current activities and some activities we are planning to do. So as you might have seen that OCNS has become a member of the International Neuroinformatics Coordinating Facility. Now our treasurer, Leonid Rubczynski, has become an elected member of the INCF Governing Board, and Sharmila Venugopal, who's our Education and Training Chair, sits on the INCF Council for Training, Science and Infrastructure. One reason we wanted to join the INCF is that the INCF hosts the Neurostars discussion forum where we now take part. And as you've seen during the conference, this Neurostars discussion forum is a very useful place to ask questions and exchange information about computational neuroscience. So we want to keep this active throughout the year. And we established a keynote lecture series in honor of our honorary member Wilfried Rall, who is one of the founding fathers of the field of computational neuroscience. So this year, the Wilfried Rall keynote lecture was given by Matt Botvinnik, as you might have seen. In the future, we want to keep an increased online presence, not just during the meeting, but also over the year. And we're currently discussing the possibility to hold CNS meetings that are not just physical meetings, but that also contain an online component. So if you have any input, that would be very much appreciated. We would also like to hear some feedback about you. So one of our plans is to run special interest groups, 6. So this could be a group of people recruited from our membership who have then a liaison on the OCNS board of directors that focus on certain themes such as infrastructure and tools, or another theme we have in mind is outreach and education. If you would like to suggest any other special interest groups, then please do so, either using the Ask a Question button or by contacting us directly. Another plan is that we would like to organize some symposia. These are extended workshops with invited speakers focusing on some areas we think are important or will become more important in the future. One of these areas is the interface between machine learning and computational neuroscience. And we'd also want to reach out more intensely to the community of experimental neuroscientists and invite some world-leading experimentalists for a future symposium that we're hoping to run at CNS next year. And then I would also like to point out that some of the directors are rotating of the boards. So we are in need of 5 new OCNS directors, and there will be a call for application for OCNS directors soon after the conference. So finally, I would like to give a brief report about the OCNF membership. So we currently have 189 faculty members, 155 postdoc members, and 266 student members, and about 2,000 inactive members who are not currently paying any membership fees. That's a small decrease compared to last year. Of course, the decrease is based on the fact that CNS 2020 online didn't charge any registration fees. So that meant that one of the main member benefits, which is reduced meeting registration fees, didn't really apply this year. As you know, as an OCNS member, you are eligible for a large number of benefits. In addition to the reduced meeting registration fees, this includes eligibility for travel awards, and some of our members are also organizing computational neuroscience schools, which receive financial supports, etc., etc. So this concludes everything I wanted to say for now. So I'd like to hand over now to our treasurer, Leonid Rubczynski, and who will give a financial report. Thank you.
All right. Thank you very much, Volker. Leonid is here. I'll kick your screen off, and Leonid can share his if you want.
Yes. Let me see if I can. Good morning, or good afternoon, or good evening, everybody. Let me see if I can share the. Oh gosh, where is this button? Here. Share screen. It should work. Just a sec. I'll do it in this way. I have 2 screen setup. That should work. This.
Probably a good idea to share the full screen. Yeah. Okay, there you go.
I think it should work now. Right.
Yeah, that's good.
Okay. Can I go forward now? Thomas, can I go ahead and talk briefly with it?
Absolutely, yeah.
Yeah, we can see it.
Okay, great, thank you. So just a brief report about our financial statement, and my apologies, I'll keep my head turned a little bit. I have a 2 monitors set up. I want to look at the numbers I'm going to talk about. So a few words about our financial data, our budget. We are doing quite well, I would say.
Yeah, okay, thank you.
Yeah, try to magnify it. So this is our budget state of affairs. There are 3 columns here: 2018 Seattle meeting, 2019 Barcelona.
Leonid, can you zoom in a bit more? People can't read it.
Okay. Is it better now?
Yeah, I think that's just about okay.
You can full screen it.
Yeah. So current year, year before, and year before that, just a couple of words about attendance. We had about 500 in Seattle, 630 people in Barcelona. This is the number which I checked last week. So apparently we have about 600 registrants more. So we're approaching 2,500, a huge number. Last week we had about 70 countries. This number might go up as well. Now, in the area of income, we have about $6,000 projections this year, obviously, because we're not charging registration fees. But otherwise, we're staying quite healthy. We ended last year, slide this down, slightly better than we expected. So we had a surplus of about $36,000. We are projecting to lose about $66,000, $67,000 this year. But these numbers are a little bit misleading, and this is related to how this accounting is done, because we've paid about $30,000 for in-person meeting in Australia as deposits to Melbourne Convention Centre and the other organizations. We cannot recover them, but thanks to the local organizers, we can keep them as a deposit for some time. So we may be able to use them in the future, so they're not quite completely lost. Other than that, and this is about $30,000. So our year cost about $35,000 with the meeting, with the publication fees and things like that. We will have a balance of $340. Given the COVID situation in the world, who knows how things will continue to develop. So chances are next year this number will continue to go down, but I think that we have a decent reserves to stay healthy as an organization for several years, even if we will need to skip another in-phase in-person meeting. So other than that, there are no any significant changes in the budgetary in the budgetary structure. Finally, I just want to say thank you to many volunteers who joined the World Organizing Committee. My name was there, but I was kind of standing on the sidelines as a treasurer, at most with them, and Thomas, the program committee chairs, who put so many hours of great work in order to make this online meeting happening. Thank you.
Thanks, Leonid. So I think, Thomas, it's your turn now, isn't it? Are there any questions about the budget, which you could, of course, post using the Ask a Question button? Sorry, I'm still getting used to the online format.
Right. So I'll go ahead and share my screen now.
Mm-hmm.
Right. And with some luck, everything going black is not a good sign. Aha, here we go. With some luck, you can see my report from the PC Share folder, the presentation now. Unfortunately, I lost the feedback, what you can see. So from my side, as I said, I'm Thomas Novotny, the current program chair for the last year, this year. And I wanted to quickly report on the paper submissions and the process. As was already said, the meeting was planned for Australia, and we were working happily along with that brief, and we had originally about 253 submissions of people who wanted to submit to Australia. Of these, 79 chose oral or featured preferred. And in the final program, we ended up with 4 featured orals, 14 orals, and 226 posters. And overall, we had only 2 rejections, which is typical for CNS, because in the posters we are not trying not to be too selective in terms of topics or anything. So we only reject things that are clearly not acceptable. And we had 7 withdrawn.
Thomas, sorry.
Yes.
Did you want to show some slides? I mean, it's fine if you don't, but we can't see any slides.
Well, that is rich, because I tried to. Okay, let me try this again then. Very strange, because I did share the screen. Try that again. It's thinking about it. All right.
Okay, that's looking better.
It works now, yes.
Right. Well, there's always a surprise, isn't it? So we couldn't see all the things I was pointing out earlier. So with some luck. Well, it's still a bit limited what it's doing here. Oh, okay. What is? Can you see your PowerPoint now? It says
yes, it works now, yes.
All right, okay, yeah. Little hiccup there. Right.
No.
So,
yeah.
Yes. Yeah, it is temperamental, I have to say. Good. So that's about the paper submissions and acceptance rates. So you see, some conferences are very proud of their acceptance rates. So if you look at it from a featured orals and orals perspective, we are quite selective conference, right? 5% or 17% acceptance. But we do let people and young people present posters as much as we can. Okay. Just to have a quick look at this, I think it's good to reflect a little bit. That's the origin countries of all authors. And as you can see, it's always a big spread, but this is an exceptional peak here. We have most Australian authors, followed tightly by the US. Of course, that had to be because we were going to Australia, right? Now I feel a bit bad about all those authors who then had to present things late at night because we did the meeting online with a certain focus on where the most members were in terms of the timings. If you want to look at by continent, I find this is quite interesting just for the joy of looking at it. So here are the last 3 meetings. We were in Seattle, we were in Barcelona, and now we were online, but planned for Australia. And you can see a similar trend that we always have. So if we go to North America, the North American papers are the most. So this is contact authors on the left, all authors on the right. And if you go to Europe, the Europeans are the most people involved. But then coming down here, this online version now with Australia planned, we see what's here called Oceania, which is mainly Australia and surrounding areas. Does pick up a lot more than normally. Yeah, so we can see from 2019 to 2020 picked up a lot. But can't beat the Europeans who are then still dominating. Typical pattern that we've seen before. Okay, that was the paper, so that's what happened there. In terms of the program committee, this is the current program committee, and these are the folks who have selected the current program and have done a lot of work in the background around reviewing of all of the papers. And you see the names there. Some of them will have to be replaced because they have been here for 3 years. And we're still looking for volunteers to join us in the program committee. And then as a last thing, I already mentioned it, this was my last year as the program chair. Now a new program chair will be Christiane Linster, and she's taking over after this conference. And as I said, we also need some more program committee members. And that's all that I had. I'm happy to answer any questions. If you post them, we can pull them up and talk about them. Okay. And then getting back into my chair role here. As next item, we have CNS 2021, and I'll invite Thomas Knöscher on screen. And I believe I'll have to let someone else go, so I'll let Leonie go in a moment.
Yeah, thanks, Leonid. Of course, I would also like to thank you for your excellent work as a treasurer. It's really good to see that our finances are in such a good condition.
Now I cut him off, I'm sorry.
He will still be able to hear it.
Yes, he will hear it, but he can't respond. Okay, I hope Thomas is coming in. Looks good. In the meantime, we have a few suggestions in the questions. We have a question about the abstract publishing. Okay.
Yeah.
And something about diversity. Let's have Thomas Knöscher first, and then get to the question I suggest.
Yeah, we've got the mascots there as well. So normally we do the mascots first and then the questions.
Yes, obviously. Okay. So
we have time.
It's connecting, but it's taking awfully long this time. wonder whether we have a problem again with too many video screens, but it shouldn't be.
You need to kick me off.
I'll remove you for a moment, okay, and I'll get you back at some time. Okay. Let's see whether that helps. No, no real progress there. I'll try again. So new invitation, Thomas.
Okay.
There we go. Okay.
Yeah, it was my fault. I had 2 of these broadcasts open.
Ah, all right. You can already share your screen if you want.
Yeah, where do I have to do that? Where was it again?
There's a button over. If you hover over yourself on your crowdcasting.
Bloop. Where is it?
There should be a share screen button appearing. No?
No, I don't see any. In which direction?
When if I'm on my own.
Okay. Share screen. Okay. Oop, that was. So share screen.
It wasn't your fault.
Hi, Thomas.
And I take that one. Hi. So.
All right, that's good. Perfect.
Great. Okay, you can see my screen now?
Yes.
Yeah.
Okay. Then, hello everybody. It's my pleasure and my honor actually to give you a short glimpse on the place where we hopefully will have our next meeting, our next OCNS meeting in 2021, and that is Leipzig. And this is this list of local organizers. My name is Thomas Knöscher. I'm heading the Brain Networks Group at the Max Planck Institute for Human, Cognitive and Brain Sciences. My colleague Christian Döller at the same institute is director of the Department of Psychology. Then we have another Max Planck Institute, the Max Planck Institute for Mathematics and the Sciences with Jürgen Joost. The University of Leipzig with Mark Schönwiesner from the Institute of Biology, and Fred Hamka from the Technical University in Chemnitz. And we are supported in technical and organizational issues by Conventus, which is a very experienced and established company in Congress and conference management in this part of Germany. And the person is Jutta Fach. So, for those of you who have a hard time to locate Leipzig, maybe there are some, it's here in the east of Germany, not very far from Berlin, actually. So you can get there by air. So this our airport is connected to all the major hubs in Germany and Europe. But there are also quite good train connections. So you can also fly into Frankfurt or into Berlin and then take the fast train. And of course, there's also the road. So what about Leipzig? Leipzig is a medium-sized city of international standards. In Germany, it's already quite a big city of 600,000 inhabitants, and it's actually very fast growing. It's I read it's the fastest growing city in Germany currently, but it is not so big as, for example, Melbourne, and that makes some things also easier if you have a conference there. If you just consider our venue, which is about here, we'll talk about that in a minute in more detail, and for example, the main station, then we have a distance of 1 kilometer. To the airport, it's 15 minutes per train. The city center, which is about that square here, houses, for example, the university, but other important buildings like the concert hall, and the opera, and from there it's also 1 kilometer only. And here, this is just to show you, this is the place where I work, the MPI for Human, Cognitive and Brain Sciences, and this is the other MPI. So everything is nicely in a radius of about maybe 1 to 2 kilometers. So about the venue. So our venue that we have chosen is a bit different from the usual convention centers that you have in every major city. Huge concrete buildings. So this is a historic building of, I would say, amazing architecture, not only outside, but also inside. So it has been built in the around 1900, and it has rooms with Jugendstil and Art Nouveau and Neo-Baroque and what you want. Nonetheless, it has been converted into a modern convention center with a large number of rooms with capacities up to 1,100 seats. And most importantly, as I've already pointed out, it's quite near to the city center and right adjacent, that's why it's called Kongresshalle am Zoo, to the lovely Zoo of Leipzig, which I really recommend to everybody. So you can hear the lions roar, actually, literally from there. So what about Leipzig? People who are not from nearby would associate Leipzig at first with music, I think. Leipzig has a rich tradition in music. Many famous composers and musicians have worked here. First of all, of course, Johann Sebastian Bach, who spent the better part of his life working at the St. Thomas Church as a cantor, but also people like Felix Mendelssohn Bartholdy, Richard Wagner, Gustav Mahler, Clara and Robert Schumann, and many others. So, but this is the past. This is also alive at present. So we have a very rich music life. We have the famous Gewandhaus Orchestra, for example, the equally famous Thomaner Choir at the St. Thomas Church. We have a rich life of music festivals, both classic and modern. Hopefully, it will be alive again next year. This year, it was a little bit cut down due to this Corona situation, but hopefully this will ameliorate. What else about Leipzig? Leipzig is one of the greenest cities in Germany. 1/3 of Leipzig is actually green area. And if you look on the satellite picture here, so this is Leipzig, this is the city center, I've marked it for you. You see this green belt right through the city, which is an area of rivers surrounded by forests and parks. And in the south, we have a large area of lakes, which are actually former open pit coal mines and now renaturated. And it's really worthwhile to explore this green lung of Leipzig using the bicycle or by boat, by canoe, or just on foot. So as you can see here, it's not only green and music. Leipzig is also a city of beautiful architecture because it hasn't been destroyed so much in the war like many other German or European cities. And there is a lot of amazing architecture from the last 2 centuries and even beyond. Here you can see, for example, this is an industrial area. Yeah, it looks like a palace, but it's a former textile mill. It's residential buildings and artist ateliers and so on. We have the buildings from Renaissance, as you can see here, the old town hall, from Baroque, from the imperial times, and the city is full of these beautiful residential buildings. It's actually making up the better part of the inner residential circle. So it's really worthwhile to walk through the city, through the streets, and have a look. Okay, and of course, if you have a conference, people are interested in what can you do, yeah, what kind of fun you can have, and there has been a saying that Leipzig is the new Berlin, meaning you have a bustling life of cultural people, of musicians, of artists, lots of restaurants and places to go. And this is also quite well developed in Leipzig, actually. People are flocking to Leipzig, and we have this rich life there. Gorgeous eating, live music everywhere, funky bars, also more serious things like art and galleries, and so on. Yeah, and with that, I would like to finish with just expressing that we look forward to seeing you in Leipzig in summer 2021, and we sincerely hope that it will be possible to have an on-site conference again. because as nicely organized as this conference was, and I really was surprised how well it went and how cool it actually was, to go to a place and just casually meet people who haven't been known before is something that cannot be replaced. And with that, I thank you for your attention. Should I?
Thanks very much, Thomas, for your nice presentation. That's great. So.
Okay.
Video. And Volker, do you want to share yours again for the master?
I will.
And who remembers the dates of the? It's already fixed, right? So.
I forgot that. I'm very sorry, and I even would have to look it up. What was the date again?
Yeah.
That is the most crucial information.
July, yeah. We'll post it as soon as we get hold of the dates. So maybe you can post it in the comments section. So yeah. I saw that we get quite a few questions. Well, the next agenda item is, as most of you who have been to several of these CNS meetings will know, is the mascots. So the CNS meeting alternates between a location in Europe and a location outside Europe now, already for the past few years. And in 2009, when the meeting was held in Berlin, that was the last time the meeting was held in Germany, the directors back then came up with the tradition that each CNS conference had a mascot that represented the location of the conference, basically. So some of these mascots have been a bear, which represented Berlin, then a bull representing San Antonio, the Swedish Dalarna horse representing Stockholm. In Atlanta, we had the Georgia peach. In Paris, the meeting was represented by a frog after the baguette, which was initially chosen, got too old. Then we had a moose in Quebec City. We had Schweik in Prague. We had Korean dancers in Jeju. We had Milou, I think is the original name, in Antwerp, and from Tintin, of course. And we had an orca to represent the conference in Seattle. And I don't actually remember what the mascot in Barcelona was. So this picture was taken when the conference was held in Barcelona last year, and then the Barcelona mascot was presented. Normally that would have been presented this year also. So this year, the Web Organizing Committee, led by Anka Dolotsmihu, organized an e-mascot competition, had several entries, and then came up with a short list of 3 mascots that were in the top 3 places. You see, interestingly, the spirit of holding the meeting initially in Australia has affected how these mascots were designed. So you see, there's definitely a bit of an Australian feel in it. And out of these 3 winning mascots, the ultimate final winning mascot is this one. So congratulations to Jessica Lopez Azaz at the Universidad Autónoma de Madrid, who has come up with this design. So I can see there's a koala there and a big brain. And if I interpret this correctly, that's a boomerang, also representing Australia. And this is going to be the e-mascot of CNS 2020 online. So thanks very much for the organizers of this competition and also for the designer of our final mascot. And with that, I'd like to come back to Thomas, and I think we should now address our questions. So, Thomas, do you want to?
Right. So Thomas Löscher, I think thank you very much for your presentation. I'll let you off the screen now.
Yes.
We're looking much forward to Leipzig.
Thank you.
Fingers crossed. Okay. Well, let's have a look at the questions then. I think we skip the SIG bit for the moment and look at other questions first. Okay. Let me see. Where do we?
You have to read this out. I mean, I can also do it.
Yeah. Well, I mean, I just going to read them out and then we can take turns.
Hmm.
Thanks for all the work. How do you address diversity across the committees and speakers? And a follow-up comment, the local committee for OCNS 2021 is male only.
Yeah, that's a good comment. That's something that has concerned us in the past. So when I looked at our presentation about Leipzig with Thomas and myself on the screen, my immediate feeling was that I should really grow a beard to fit into the triumvirate of male German organizers. But we have always actively tried to get women, female members involved in OCNS. So a couple of years ago, the president, Astrid Prinz, was female, and the treasurer, Victoria Booth, was female, and she was preceded by Frances Skinner, who was a female treasurer, and of course we had Ranu Jung, who was a female president. And
so I can also say a few words. I mean,
yes, please.
It's not only the past, right? So we have taken great care also that actually the new program chair is female. This was deliberate, yeah, and it's not kind of a coincidence. And if you look at our committees, we are always encouraging not only the gender balance, but balance in all kinds of ways. So the board and the program committee are quite balanced in terms of gender. In addition, we look at geographical balance, if you want to call it race, yeah, you may. And even in the submissions and the oral presentations, we always look at that. And we have done so far every year that we promoted some talks to increase the diversity of the speakers. So I think we're trying hard. There's always more we can do and try to improve. And if you have specific suggestions what to do differently, and we are always very happy to consider them. But in the background, we are actually trying quite hard to balance things out. So please come back with in the chat or with a comment on the question if you want to add, or if you want to join on screen. I'm happy to do as well.
I mean, maybe to add to that, something else we have considered is also whether we should be more active in terms of keeping a record of ethnicity, gender, etc., of all applicants, right? So there's always a balance with data protection regulations, but maybe we need to be more active on that front. So Thomas and I already had an initial conversation about that. Diversity SIG. Right. Interesting idea. Okay.
Okay. I'll move on to another question for now. I don't see anything happening in the chat here about this answer. So the other question I've picked out here is why does abstract publishing cost so much? And I can say a few words about that, and Volker can too, obviously. But basically, what the cost for abstract publishing is that we are paying the publisher, which is a BMC Neuroscience, per abstract a publication fee, and that's the cost basically. And unfortunately, publishing is expensive. So if we publish some, it's about 250 abstracts this year. With a charge per abstract, then that adds up to that number. That's the answer. I mean, there are other publishers, but they're not necessarily cheaper. No? If you try Frontiers or something, it becomes more expensive.
Yeah, we compared different prices, and there's not really a big difference. So we'd be very happy to pass on that question to Springer Nature, who are now publishing BMC Neuroscience. So.
Yeah, and then here there come some comments actually towards the question. I should mention those. Yeah, so Fleur, for example, has commented after Tom pointed out that maybe we don't need publication, and says Bernstein is not doing external journals. Now Leonid answered that anecdotal evidence says that many people do think it's valuable, but that can be discussed. I personally think it has some value. You can collect citations on it, and you have some sort of record of having contributed to the conference, and it's not in some program book that no one will ever look at again, which I think is the sad truth if you don't go down the publishing route. Okay, now with all these 6 in there, it's a bit hard to see what other general questions there were.
There's a question about have we addressed it? I like the idea of having an educational component. The order keeps changing. E.g., network modeling for undergraduate students. They are the next generation of neuroscientists, and having this experience may encourage them to stay and contribute to the field for years to come. By Annalisa Skimemi. Yeah, Ankur has answered that the outreach SIG could work on this. I think that's right. So I think we I think it's a good point, and if the outreach SIG could address that, that'd be fantastic.
Yes. Now I'm scrolling through. I see. Yes, so I say this is answered. So the rest is 6 suggestions, I believe.
Yeah. So if you have any other questions, then please post them now, and I think otherwise we can probably conclude this meeting. So
yeah, just to say a couple of words there. They're bubbling up with a few votes, but we have about 103 people in the room, and I count on the order of 15 votes here. I think we have to continue this offline, and we could do a bit on Eurostars as we are at it, and see what people think. And otherwise, the board will discuss it as well on their annual meeting.
Yeah, it would be definitely good to add this as an item to Eurostars. Ankur, and I think Soledad has been adding these to Eurostars, right? Would it be possible to add a Eurostars item about the member meeting where you can continue the discussion? Then we can, I mean, this education and diversity are 2 points that can continuously come up, right? So this is definitely something we need to carry on discussing. And
it is always an issue, and of course, it's not an easy issue.
Thank you. Okay, thanks, Soledad and Ankur. And yeah, on a personal note, I'm very sad to see Thomas go as a program chair. So I've worked closely with Thomas for many years, and I have to say he's always done an absolutely amazing job. So I'm very happy that Christiane is taking over, but I'm also very, very sad to see Thomas go. So I would really like to thank him very much for doing such a brilliant job. Thank you.
Thank you very much, Volker. Now we have a late-breaking question here, and I thought, why not? And I'm putting Tom on screen here real quick. With some luck, it will work. Oh, there he is.
Hi.
Do you want to just quickly say?
Yeah, just curious about the Leipzig conference location, and if there's going to be a timeline for maybe when a decision would be made as to if that converts to an online event.
Could you speak up a bit, Tom? It's very difficult to hear you.
Oh, sorry. I'll just talk a bit louder. Is this better?
Yes.
Yes.
Okay. I'm just wondering for the Leipzig event, if there will be some time at which it's decided to go ahead with the physical conference or convert it to an online conference in the event that it's not possible for some reasons.
Yeah, I mean, we're obviously monitoring the situation, right? So we can't say at the moment exactly what is going to happen. So we're currently discussing the possibility of having a hybrid meeting in any case, whatever the COVID situation would be, right? Because we, of course, like many people here, we noticed that this online conference worked extremely well. So, and a hybrid meeting would give us the possibility to invite speakers from all over the world who might not be able to travel to Leipzig, whatever the situation. But I can't give you a date right now. So.
Okay, that's all right. Thanks very much.
Thanks, Tom. And thanks again for helping with the organization.
Okay.
Cheers. Yeah, so we'll keep you posted, I guess, is the answer. And we'll have to play it by the ear. Okay, thank you very much, everybody. I think, yeah, Volker,
Yeah.
shall we call it closed?
Yeah, let's close the meeting and enjoy the rest of the conference, everyone. And thanks for everybody's contribution. And obviously, everybody here is helping to make this meeting a success. So I would like to thank everybody. And if you're not a member yet, then please do consider becoming one.
Okay, thank you very much.
Bye.
Bye.